Thank you. Go ahead,
So,
Mr. Rakesh.
okay, so I'm a plant breeder by training.
Okay.
And we are having our colleagues with me, and I can go ahead with that. Other people can introduce themselves. Dr. Professor Namo.
Good morning, good afternoon. My name is Namo. I work as an agronomist here at the International Center for Biosaline Agriculture. Thank you.
So he's agronomist.
Now, nice to meet you. Welcome.
Yes, Dr. Hibzur.
Good afternoon. Good morning over there. Actually, I am Hibzur Rahman, working here as a plant molecular biologist and looking after the Desert Life Sciences Laboratory.
Okay, thank you. Nice to meet you. Okay, on behalf of INTA.
Over to you.
Yes. I introducing myself and then I pass the floor to the other colleagues. My name is Guillermo Sanchez. I am the manager of institutional affairs of INTA. Silvana, may you introduce yourself? Are you muted?
My name is Silvana Fancio, is communication in office the institutional, the INTA.
Gustavo.
Oh, good morning. My name is Gustavo Pasten. I'm part of the Institutional Affairs Office. Okay,
thank you, Gustavo, Silvana. We are the team of the institutional affairs of INTA, and now I pass the floor to the professional, the experts in the topic that we will try today. Luis Erazo and team.
Good morning. I am Luis Erazu. I am a national coordinator of industrial crops at INTA.
Hello, I am Luciana Martinez Carcina, and I am a researcher in Farmacia Research Station of INTA.
Good morning, I am Paola Fontana. I am a coordinator of the genetic improvement of industrial crops.
Thank you. Diego.
Good morning, everyone. My name is Diego Ortiz. I'm in charge of the sorghum breeding program, and I'm also in charge of the national plant breeding network in INTA.
Okay. As surely you remember, in my email, I introduced a brief agenda to be developed tomorrow. This morning, sorry.
Yes,
the first point in the agenda is related to quinoa crops. Yes. So my suggestion is to pass the floor to the industrial crops team in order to explain some of our capability regarding these crops. So I don't know, Luciana or Paola.
Okay. Yes, may I share my screen?
Yes.
Okay.
Lo puedes hacer, tenés para.
Permiso.
Ahí ya, ¿a quién le pasó? ¿A Luis? Sí, estás como co-host, Luis.
Sí. No, a ver. No puedo compartir.
A ver.
Hacemos como
Dale, pasale el host directamente.
Sí, ahí fue. Erazo. Doctor Erazo, excelente.
A ver ahora, Luis, si puedes.
Acá debería aparecer. Salvo para
No. Hasta el momento no. A ver, intentemos ahora. Le pasemos a un email. Pasarle a que tampoco lo puedo bajar.
Si no, pueden pasármelo por correo y lo paso yo de acá.
Bueno.
it's really big, the file, so I don't know if by mail it's possible.
Okay.
Está en silencio, doctor.
Luis, ¿tenés el botoncito verde para compartir Share Screen?
Sí. Ahí está.
Bueno.
Ahí está. No, recién me habilitó.
Ok. Okay.
Gracias.
Delay of the system. I'm sorry. Ok. Tendrías que ponerlo en el modo presentación. Ahí te...
Sí. Tiene un delay ahí, así que...
Pero vale. Saquemos la imagen.
Ahí está perfecto. Ahí estamos bien. Mr. Rakesh, can you see the screen of Luis?
Yes, we can see that very well.
Okay, thank you.
Okay, now here in some slides, we will summarize some of our capabilities regarding quinoa crops. Here in INTA, we do have a germplasm bank. As you can see here in this map, all these numbers represent some collection sites from where we do have some 45 sections. Here is after some genetic studies, we realized that these different collection sites, they do have genetic differences between the origin, just like dry valleys, humid valleys, transition area, and puna. So this variability allows us to work in our quinoa genetic breeding program. Paola Fontana, she coordinates that program. Here there is a map where we do have some of our research stations. Here we do have, as a part of our genetic breeding program, we do have a cultivars assessment network. All these points with the red arrow are some sites from the northwestern part of the country, the central western part of the country, and also we do have some sites in the southeast of the country. All this brings us a lot of agroecological variability in order to assess or to test our promising lines. And as a product of our genetic breeding program, we released a cultivar Hornillos INTA Jujuy government. The origin of this line is the highlands of Argentina, of the northwestern part of Argentina, but it's very well adapted to other agroecological regions. And also, now we are releasing a new variety called Morrillos INTA. The origin of this one are lowlands, but it is also very well adapted to other sites here in Argentina. Also, we are at the final stage of evaluation for registration of 4 other promising lines. Regarding quinoa crop management, we do have some ecophysiological studies, such as sowing dates, plant densities, and also we are studying we are doing some studies about plant nutrition for conventional systems, but especially for organic farming. Also, as a strategy for adding value, we developed post-harvest machinery. Here you can see a threshing machine, specially designed for small-scale producers, and this reduces a lot time of work for farmers. We also developed the quinoa cleaning and classifying machine and deponifier machine. Here you can see some photos of trainings that we are doing as part of the projects that we have with communities, with a special focus on youth and gender. Here we train people about how to clean and to cook these quinoa grains and how to introduce them at the family diet or food. And also this is the final stage of other trainings about crop management and some varieties selection. So here for us it's very important to work with communities and to add value to this product that they produce in their farms. We also did some technical cooperation to Egypt, not only for some field studies. You can see here in this photo, these are assessments we did in Egypt in order to study adaptation of these quinoa varieties at those agroecological sites. But also we trained some women and some students and a lot of producers. So we had a very nice experience with Egypt. We published some papers and also we are still working together in some other issues. And then we had some technical cooperation to Morocco that is still on. And here we also worked at field and also at the University Hassan too. So we are doing a very interesting work here with Morocco's people. Here we left some of our papers that we wrote. There are some more, but here are what we think they are more interesting for you. And well, any other question? Here are our contacts. Thank you very much.
Thank you. Okay.
Thank you very much.
We are open to your question. I don't know if you want to do some presentation regarding
Yeah, I
this
will do
topic.
that presentation. But before that, I would like to know a few things, particularly. So your breeding program is based on the recombination breeding. Are you doing or mutation breeding, or what kind of breeding program you are pursuing? Is it
Yes,
by making some crosses?
Conventional breeding program.
Hmm?
Yes, we do have a conventional breeding program.
So no, I mean to say, is it from the selections or mutation, or are you making the crosses and developing some different lines?
Creo que está preguntando si usan mutaciones inducidas o algún tipo de selección de líneas.
In fact, we have a high variability in the accession that we collected years ago. And we select in the accession some genotypes or phenotypes, and then we go ahead to select some interesting lines, and we now are introduce mutation tools for generate, yes, for increase variability.
Okay. So because I saw that your Horin loss and Mori loss the accessions when you showed us in the picture, and you said that it is a variety, but I saw the like a different kind. So that's why my intention was why they are having the different phenotype in the same field, or is it changing the color depending upon the maturity? So
no. Yes, the color is different because we have variability for color, but they are not related. These two lines, Hornillos and Morillos, are not related, and it's not the maturity level difference in color. It's just that they are not hybrids. You have variability in the selected lines. I don't know.
Within the line, within
Yes,
like
within the line.
Hornillos, within the line.
Yes.
Okay. And also about the desaponification machines you have. So how costly it is, and then do you have something like, because you worked in the Morocco and Egypt, did you transfer the technology to different country, or how can we take the help or like, the advantage from you on desaponification machine?
Yes, INTA developed the desaponifier machine, but there is a private group that is producing it, so we do not know the price of this machine, but we can answer and then answer to you because we don't know the price.
Okay, thank you. That would be great.
Okay, thank you.
And then in Morocco, if I am correct, you are working with UM6P?
In Morocco, we are working with Dr. Wafe Ben Labib at the Hassan II University.
Hassan II, okay.
At Rabat.
Okay,
but
okay.
we did have some trials at other sites of Morocco and the West.
And in Egypt, is it from the DRC with DRC or somebody else? Egypt. Working with Egypt with DRC, Desert Rice.
En Egipto, ¿con la RC de Egipto o con quién?
No, with Ain Shams
University.
Ain Shams University.
Yes.
In Sham.
We have an MOU with that university.
In Sham.
Yes.
Okay. Because why I'm asking, because we also have the collaboration with Egypt and Morocco, etc. So we want to know that whether it's the same or different.
We regarding the ARC that you mentioned, we are working in some topic to work together with the ARC, but we are in the first exchanges with the ARC. But the university, we have an MOU long time ago.
Okay, and then last question on your presentation. Do you have some varieties which are sweet quinoa without saponin free?
No, we know that there have been a lot of work about that, but they have a lot of problem with pests. So we just breed some saponin varieties, high level or middle. Morillos has an intermediate level of saponin, but not sweet quinoa.
Okay. Good. Okay, so then I can share my screen and I can do the presentation for ICBA, what we are doing on like latest in the quinoa, if it is okay with you.
Can you do the presentation?
Yeah, I can do the presentation for quinoa, but the latest what we are doing.
Okay.
Can you see the full screen or?
Yes.
Or a half screen?
The half screen.
Okay, so let
we
me
are
shift.
we are seeing your
Slide
presenter
show.
screen.
Yeah. Let me switch.
We can see the slide clearly, even though in the half screen.
I think this is better. Full screen now?
Yes.
Yes.
Now it's full screen.
Yeah, okay. So thank you. So this is about the ICBA work, which we did it we are doing from 2006 to onward. And we got something like more than 1,000 Quinoa accession from the different research facilities. And then we tried to evaluate them at our station. We found that some are good up to the yielding up to the 4 tons per hectare. And then from there, we find out that 5 of the lines were good, with a stable yield. And this was also done, as you mentioned, about the mass selection method from the accessions, which were good on that. Then we look for the multi-locational testing, because once you are having the good view, you need to know whether they are also good at other places. So we just find out whether they are good or not, and promising multi-purpose type, which is also having the grain and fodder both, because in the marginal conditions, we need the fodder also. And then we had the outreach program where we just extend our research to the different countries: Yemen, Egypt, Jordan, Oman, Morocco, Spain, Uzbekistan, Tajikistan, and Kyrgyzstan, with the support of donors. And in 2016, we organized one international conference on quinoa. I think somebody from Argentina might have also participated, but I don't know, because I joined in 2018. But before this conference very successful conference was convened, and there were more than 150 delegates from 48 countries. 2019, when I joined, then we discussed that how we can work more on the quinoa genomics, so that we work on the quinoa genomics at ICBA, and we try to develop some molecular marker using the GWAS technology in the recombination breeding program. And then we started in 2020 for the recombination breeding program. And how did we do that? Because once we do the recombination breeding program, it means we need to know what is the target. What is the target? What is the target means what type of plant you need. And that way we can say that's called product profile. This is one example of product profile. So this is for the MENA region, Middle East and Northern Africa. What should be my target when we are growing for the breeding program? These are the targets which we made for the must traits, range traits, and win traits. So that's why I was asking about saponin free. It's a winning trait. It is a game changer if we can have that type of thing. So in this one, we started working with our one molecular lab. We developed that, and then it is inaugurated by our minister, and now we have this lab fully functional, where we are working on the molecular markers. And then since we have the quinoa genome sequenced, it's by another study from the Jarvis et al. So we started working to take the advantage of that complete sequence of that quinoa, and we try to develop our mapping panels. So when we talk about the QTL identification, we need to have something mapping populations. So the mapping populations, we develop the mapping population using the association mapping panel. Because if we are developing biparental, it takes time to develop the mapping population. Or multiparental magic or nested associated mapping, it takes time. But association mapping panel you can utilize using the accessions which are diverse and not very related. So we use that one, and through that we developed a association mapping panel. Using the polymorphic markers, we find out that which are good and different genotypes, or little bit different, So these are the things which we need for QTL identification. We need the mapping population. We need the polymorphic marker so that we can distinguish different parents. So this is the mapping panel, association GWAS panel. And it was for the different traits, and we tried to incorporate the extremes as well as the intermediaries, so that for the different traits, so that we can have the good diverse population. And even in the saponin, you can see it's a high, low, and free of the saponin. So this is the one which we took from low saponin as well as the high saponin. So we took the genotype from this extreme to the lowest extreme, and also the middle one, so that we took the all things in the our genome mapping population. And now it is almost ready, and we are working on the GWAS study, genome-wide association mapping. So this around 190 genotypes are resequenced. Dr. Hifzur is already here. He will explain in case if you want more. Then we did the phenotyping, because phenotyping is the key. Genotyping you can do, but phenotyping is the key. When you are doing the phenotyping, you have to look for that different traits, whether they are good or not, and what they are, how they are performing at different locations. Whether it's a yield, whether it's a plant color, or this inflorescence color, or maybe the height, the flowering, everything. So we did it at our station in 2 seasons, as well as the different countries. You can see that it's from the Bhutan, from Togo, from DRC, this Africa, and this Egypt, then in Mozambique, also in ICBA, and also in the Central Asia. And using that, we tried to put a genotypic and phenotypic correlation, that's called marker trait association, which is going on now. And in the meantime, we also developed some recombination breeding program, where we have developed the crosses from the different parents. And that's why I was asking whether you are also doing the like a recombination breeding or not. So we started work on the recombination breeding in 2020-21, and that way we just made the different crosses, and those crosses were tested. Once we made the crosses, it's not only the crosses, because you may feel that this is cross, but it may be self. So we try to develop the marker system where we know the hybridity. So the first, to test whether they are hybrid or not, you have to find out the parental polymorphism, the parents which were utilized in the crossing purpose. So those crossing purpose parents, we just find out these are the different parents which are like a distinguishing from one to another using the specific polymorphic markers. And after that, we found, okay, these polymorphic markers will display whether this cross which is made F1 is actual or true hybrid or not. That is the reason. And after that, we are now working on the shuttle breeding. Shuttle breeding means once we do the generation advancement at here, the second generation we grow in the Central Asia, our another office in Central Asia. So the Central Asia is a different climate. So we can have the good genotype, a generation advancement in the off season, and also stability of the genotype, which is more widely adapted. So that's called shuttle breeding. Then molecular breeding, we are working on the, as I mentioned, about the crossing recombination breeding. And the third one, which we are trying to do the fast track our breeding program, is through the speed breeding. In that, we are trying to develop some containers with the photosensitivity, means photolights, so that we can grow quinoa at least 3 to 4 times in a year, so we can have the generation advancement. And within 1 or 2 years, we can develop the variety through recombination breeding. The most big problem is that the society is like becoming aware, but not yet aware. Still more awareness is needed from this side of the world. Definitely in the Latin America and Southern America, the people know about the quinoa, but in the other part of the world, not many people know about the quinoa. And even if they know, they feel that this is a very rich people food, the food for the affluent people, not for the poor people. And that's why in the market also, because of less availability, it's having the very high cost, like $5 to $30 per kg in supermarket, depending upon the demand and the packaging. But definitely it can be a nutritional security driver, nutritional and food security driver in the marginal environment. And ICBA is working very, like, like trying hard to make it as a introduction as a food for the marginal environment where others cannot grow. Like those places where there is high salinity, there is less water or more like less temperature, we can grow quinoa over there, which is not like replacing wheat and rice, but at least it can work on the marginal environment so that it can drive food and nutritional security. So this is what in the Morocco quinoa value chain, you can see that we have developed the things with the UM6P. This Mohammed VI Polytechnic University. So this is the IDRC funded project and where we developed the quinoa processing machines as well as the quinoa products. And they developed some quinoa, you can say the cooking programs, cooking fairs where people come up with the different dishes or delicacies, and then they find out, okay, this is the best one, and they were awarded with the ladies, so that women empowerment was also there. So we developed this quinoa value chain in Morocco. In Kyrgyzstan, in Central Asia also, some of the farmers, they just grew quinoa and then it is already working over there and coming up in those areas because of the cold. And in the MENA region, in our quinoa, in Egypt also, they developed the quinoa biscuit, quinoa condiments, quinoa olive and fruit paste. So this is the value chain is being developed along with the mechanization. So these quinoa varieties are there. So our take-home message in this one is that quinoa tolerance to high salinity, drought, and low temperature makes it an ideal crop for marginal environments. So that way, we see quinoa is a big feature in this area, which is marred by the marginality or biophysical marginality. It does not compete with other crops. It can grow where other crops fails to grow. Urgent need to develop improved varieties in progress with desired traits. Molecular markers and speed breeding could speed up the process. This is the key. In case if we can go for the molecular marker, we need not to wait for one varieties to grow until maturity to find out which trait it has. Then needs processing, hence private entrepreneurs for a value addition can be instrumental for acceptance of society. This is very, very important. We have to have the different players in the society, then it can be accepted in the society very quickly. Awareness is required to popularize it and could be a strong driver for hidden hunger and nutritional security because of the nutritional dense characteristic of its grain. So these are the different our stalwarts for the quinoa who developed the things like Nanduri Rao, he is retired. Dr. Redwan, he is working still as a consultant in Morocco. So Dr. Mohammad Shahid, Prashant Vikram, the breeder, Dr. Hifzur Rahman, he is already attending. So he is the one who work on the molecular work. Dr. Hendash, he worked on the Egypt and Morocco. And Juan Pablo, he was from maybe Bolivia. Yes, he is from Bolivia. So he worked as a postdoc in ICBA. With this, thank you very much for my presentation. This is our institute. So you are most welcome on that. So I'm happy to answer the questions in case if you have any.
Thank you, Rakesh, for your very interesting and clear presentation.
Yes.
I don't know.
Some questions.
Yeah,
please.
yes, go ahead.
Yes. Thank you very much, Dr. Singh. Very interesting. You have a lot of work in quinoa. We just wanted to know how many hectares do you have of quinoa in Emirates?
How many accessions?
No. How many land?
The cultivated surface, the hectares.
In the cultivated area, actually, if you see that this is the UAE, it is all sand. So we are growing in the different small pockets with the drip irrigation. So if you go it with the several thousand hectares, it's not like that. It's a very sporadic, but we are having, because our mandate is not only UAE. So we have already spread in several hundred hectares in the Morocco and Egypt and Central Asia. So these are the major area where we have the expansion of the quinoa in the acreage. While in the UAE, it is sporadic with the different one field to field. But yeah.
very much. Another question. We saw you are working with some low saponine level quinoa grains. How are you going to manage pests and all those? Yes, especially with birds at maturity.
Yes, correct. So actually, in this area, even the bitter quinoa bird don't leave. So what to say of sweet? Even the bitter also, they eat everything, because here there is a less food in the desert. So even bitter also they eat everything. So sweet one is definitely affected much, but we are using in the large area, we are using the nets.
Okay.
But that is, you are right, that is not feasible when it is going to the farmer's field in the large area.
Okay. Okay, thank you. Thank you very much.
Okay, thank you. It was very interesting, the analysis of the market that you did, you shown. I don't know if which is the situation in Argentina, Luis, regarding the market, because they are showing a developed value change, high added value of product. Here in our region is similar or not?
No, in Argentina, we have a different situation. We have a different products, and like milk, for example, and milk of quinoa, and flour of quinoa, and the some demands of grains. This is the situation of Argentina. We don't have a lot of products developed from quinoa.
Okay. And also we are exporting products. ¿Estamos exportando algún tipo de producto?
No. We import some quinoa, in fact, in Argentina.
So you are importing? I was thinking that it's a quinoa producing country, actually.
Yes, we produce a lot of quinoa, but demand is so high because people now is aware and they want to consume quinoa grain. So demand is so high that we do have to import some quinoa from Bolivia and Peru, because quinoa produced here in Argentina is not enough.
Okay.
Can I ask a question?
Yes, please.
Yes, go ahead, Diego.
Thank you very much, Dr. Singh, for your presentation. It was very impressive. I was interested in the marker system that you're using. Can you explain which kind of markers?
Yeah, I will leave this floor to Dr. Hifzur Rahman. Actually, what we are doing, this is in the infancy. We started, as you saw that, we started in 1920. So we are already in the Jivas. So Jivas, it is already re-sequenced, and we are targeting, because we have already phenotyped. So we are targeting, after resequencing, we will get definitely something, the locus like a TSARL or something for saponin free or height or color of that. Once we get that, we will be utilizing that in our breeding program, marker state selections, must. Hifzur, you want to add something that?
Yes. My second question was that regarding if you have some validated markers that you're currently using.
Yeah, because this is only just 2 years back, so we are in the process of that, and actually this is something like a de novo, because nobody has done so far, and we are trying to use this thing. And I think if keeping the finger crossed, if it works well, then I think this could be a game changer for the breeding purpose.
I don't know if there is a reference genome in quinoa.
Yes, we have that, and that's what I'm telling. This in 2017, Jarvis et al. They sequenced the quinoa, and we have the reference genome. That's why we resequenced that, and we are going to align with that. Hifzur, you want to elaborate more?
Yeah, doctor, actually there has been a couple of reference genome. One has been already published from Japanese group, and other one has been published by Mark Tester Group from King Abdullah University of Science and Technology. And regarding your second question, we are using actually indel markers, what we have developed at our own laboratory from our initial sequencing, but they are not any trait specific at this particular point of moment. I cannot say that they are associated with any specific traits, but yes, we are doing the GWAS study. So from that, we will be surely able to develop some of the indel markers which are associated with trait of interest. Sorry, maybe my voice might be a little disturbing because I am driving as my son got severely sick. I need to carry it to hospital.
Okay, you
So,
go ahead.
No, I am in the car. I am attending the meeting actually.
No, you can go ahead. That is more important. I can handle that. So
no,
mostly
I am already here. I will be attending the meeting. I'm not going to home. They will be coming down. I will carry them to the hospital. Yeah,
Okay.
so that was regarding the marker assisted selection. So marker assisted selection is still we are not able to do, but yes, since we were there till now, we were doing identification of hybrids. So we have developed the markers, which Dr. RK has not presented. With those markers, we were able to select the hybrids, which were found to be positive between the crosses, what we have done till now.
Yeah, so for the hybridity testing, we are using the indel markers.
Indel markers, okay.
Yeah, for hybridity testing, but the SNP markers, I am looking forward for SNP markers. So it's SNP based, we can have that, and then we can develop from those SNP markers, we can develop the panel also later on. So
Okay.
that is in the offing. That is, we are trying for that.
Thank you very much.
Okay, thank you, Rakesh. I think that we can leave to our team to review the presentation and probably later to identify some points for a joint project. No sé, salvo que ustedes tengan alguna demanda puntual ahora. No, bueno. Yes, I mentioned that. I don't know if you, Rakesh, you are thinking in a specific point to go into to think in a specific project.
Yeah.
If not, you can leave to review the presentation of both parts, and after that to do a proposal in a topic of interest of both parts in order to exchange experience or probably a professional exchange, and I don't know.
Correct.
Yeah. Are you agree, Rakesh?
Yeah. I'm okay, but I want to just ask Professor Namo also, in case if you want to say something about, because as you mentioned, you are doing something on the showing that density and nutrition and post-harvest, etc. Dr. Namo is also working on the quinoa for those things. Dr. Namo?
Thanks very much. Thanks very much for both presentations, both from colleagues and from Dr. RK. So yes, we have started work on some of the nutrition, the fertilization program, and I was impressed to know that colleagues are working on fertilization with reference to organic farming, which is our interest as well. We would want a scenario where we combine mineral fertilizers and organic fertilizers with a lot more bias towards organic fertilizers because of the sustainability issues, CO2 emissions, soil health in quinoa systems. So we just at the beginning stages where we are trying a couple of things, including green manuring and green manuring with fertilizer and a range of green manures that can support quinoa. So in addition to that, we also are trying, or we've started trying intercropping with other leguminous crops, which we are very optimistic that we may get some inroads into developing systems around quinoa which then we can fertilize soils and get more yields. We understand that quinoa is a crop that needs a lot of support with nitrogen. That's why our focus is on legumes. So it's very interesting. Thank you very much for giving me this opportunity. We are keen here at ICBA to work closely together with you in developing management practices. But as you may know, we focus on the marginal areas. But with quinoa, I know that even in favorable areas, we may not have a lot of options for farmers. So that's an area of interest with regards to crop management. Thank you very much.
Thank you. So
Thank you, Professor Ram.
Yeah, yours, yeah.
Paola, Luciana, Luis, some question? ¿Alguna pregunta?
No, it's okay. Thank you, Professor Namo. It's very interesting. And yes, we can work together with that because for us, it must be our farmers do not want to use some chemicals. So for small scale farmers, we do have to develop. So we are already doing organic farming, but we are increasing our tests because small scale farmers do not want to use chemicals. And yes, we do have some conventional systems for larger scale. So thank you very much. Yes, we are very interested in working with that with you.
Okay, I have one more question, Dr. Rakesh.
Yes,
or Professor Ramos, I don't know. You mentioned the developing of the value chain of quinoa, and you shown the quinoa as a high price product. Are you thinking, and also you mentioned that it's a good crop for marginal areas. Do you think that quinoa is a tool for food security in those regions? I think in low-income people in our country, and also the second question is which are the scale or the size of the farmer that produce quinoa in the countries that you are working?
Okay, so first about the marginal environment and quinoa. Like quinoa is a unique crop because it is facultative halophyte. So in the saline area where you don't have much fresh water, you can grow quinoa up to the 12 to 15 decisimum with economic return, where the wheat and rice and maize, they cannot grow. So it is a marginal environment, and it is not replacing, it is not asking the people not to grow rice or something. So those areas where you don't grow anything, you can grow quinoa. Second thing is that it takes half of the water which is required by wheat and barley because it's a cool season crop. So half of the water, I'm not comparing with the rice, rice is a water guzzler. So those crops, half of that you can grow quinoa. So again, that one. So in the cooler environment and less like a drier environment, marginal soils, saline water and saline soils, you can grow quinoa, where other cannot. So that's why we can say that it is a one crop which can grow with, like if you talk about the grain crop, it's the most tolerant is barley, which is having the threshold salinity of 8. Quinoa has threshold salinity of around 12. So it's a much more tolerant crop, and I did not find any other crop as a grain crop which is more tolerant than quinoa. Grain crop. I'm not talking about the forage, and the forage can be there, but grain crops. So that way I can say in the marginal environment, it could be a driver for the food security, but not only food security, but nutritional security because of the high protein and high quality of the protein, because it has the high lysine, which is equivalent to the any animal protein. Because lysine usually the vegetarian diets, it's less. You have people say that you have to supplement with animal protein. I'm a vegetarian. So in those cases, the lysine, high lysine, which is equivalent to any animal protein, it is in the quinoa. High protein is there. Then those people who are gluten intolerance, anyway, the gluten free is there. The another thing is that I saw the people, they're diabetic, type 2 diabetic. My friend is there. He wants to eat rice, but they say don't eat rice because of diabetic. And now the quinoa in the supermarket here, they are selling the quinoa 50-50 with the rice and quinoa, because quinoa doesn't have its own taste. So they are mixing quinoa with the rice, and then the glycemic index, which is a indicator of the if you can eat in the diabetic or not, it comes down from 77 to 50-53. And that way, the people who are having the diabetes, they can eat the rice with the 50% quinoa, 50% rice, mix it and then cook it together. So that way it is good for the people who are diabetic, gluten intolerant, and it is nutritive also. And it is supporting the not only carbohydrate, but with the rice, if it is quinoa, it is more on that. Oh yeah, second question you asked.
The size of the farmer.
Size of the farmer.
Size of the farmer.
Size of the farmer here in the UAE, it's very small, but in the Morocco, it is up to the like 10 hectares, 5 hectares, 3 hectares. So in the Morocco, they have the like 3 to 5 hectares, even 1 hectare to 10 hectares. So it depends upon because some farmers are seed producers now.
Okay, thank you. I think that the size of the farmer is similar here in Argentina. Is that right?
Yes, mostly yes, 2, 3, or 5 hectares is a normal size of small farmers, but there are some experiences with higher scale systems.
Okay.
Also here.
Okay.
Oh, okay. In order to close the first point of the meeting, I was said before, we will prepare a summary of the meeting and we'll share with all the person in the meeting, in this meeting, in order to identify 1 or 2 points in that could be offset of a common project, of a joint project. I think that we need to work in a concept note in order to have the idea of the project. With this idea, we can serve financial support of the project, because we have activity developed within INTA for the institutional portfolio of project, but in this period we are working in a new portfolio, so we have no activity or the activities are ending in this time. We hope to start with a new portfolio at the end of this year. Probably some kind of financial support considering activities that each of the institutions are currently carry out in the institution. On other projects, we can search together external financial support.
Yeah, correct. We can look for that, and then we can also add on that when the draft is there, we can add where we can work together.
Okay, so my suggestion now is to pass to the second point mentioned by Dr. Sarkinai. The second point says what other food crops could be used other than staple crops for harsh marginal environment, considering something if could be matched with international year of mileage. So Diego is working in the improvement program of sorghum. So I don't know, Rakesh, if you want to explain or to point it out the topic.
Yeah. Okay, before we go that, I can ask one thing in the quinoa related because it comes to my mind to Luis also and the researcher. Do you have any accession which is having the dormancy?
No, we don't have a accession with this trait.
Okay. Okay, thank you. Okay, so about that, the second point, actually, these are the 2 points I gave to Darkanai. The second point is particularly when, like quinoa is a cool season crop. But what about in the hot season, the drier season? Do we have, because in the rice is a hot season crop, summer season crop, what, or like you can say the rainy season, summer season crop, what we can grow other than rice in those type of condition, or other than the some staple crop like wheat, where there is not much salinity, but we have the driest area and the harsh environments. What are the other crops than the staple crop? That's why I said, do we have some millets? Sorghum is there, but do we have something on the minor millets? Because they are much more strong and you can say much more robust under the climate regime, like a climate danger conditions. And they are more climate resilient, I will say.
Yes, Dr. Singh, here in Argentina and all the origin area of quinoa, we crop quinoa at the summer. In the high altitude areas, we do it just in the summer, and here in the lowlands, we do it just more in autumn, and we end the cycle in winter. So here, yes, we do use quinoa in the summer, if that was the answer, yes?
Yeah, what else than the quinoa? I mean to say, as a minor millets or something. What else?
What else apart from quinoa?
Yes.
In saline soils? Are you
yeah, I can tell you.
soils?
I can tell you. Say for example, there are certain crops like finger millets, foxtail millet, proso millet. These are the crops which can be grown very easily with the least amount of resources under the marginal environment, and they are very, except finger millet, they are very short duration crop. The crop which, you grow one season of quinoa, you can grow 2 seasons of proso millet and foxtail millet. So that way I was asking what kind of other crops you can grow in that. And since this is the International Year of Millet, that's why I mentioned 2022, it is International Year of Millet. So can we do something for the millets on that? Yes, sweet sorghum is there, I know, but what else than that? Because sorghum is also good, but sorghum, if I am correct that Diego, it is taking a little bit more water than the millets, minor millets.
Yeah, sure. Normally, there's a lot of programs in other countries that work together, the millets and sorghum. In our institution, the sorghum program works on its own. It's not related to millets.
Okay.
Correct me, I don't know, Luis, if we work on millets in the forage program. I don't remember if we have some like a general program for several grasses, but I don't remember that we have some millet program in particular.
No, millets, not in forage program.
Okay, so unfortunately we don't have some particular efforts in millets, but we do have in sorghum.
So that way, in case if you want, we can think of working together on the millet, because you can at least try that. If you have not done so far, you can try that, the finger millets, particularly when there is a drought and heat. You can grow something like a finger millet, proso millet, Amaranth also. Amaranth must be there, isn't it?
Yeah, Amaranth, there's some efforts.
Yeah, Amaranth must be there because of because it is a more on the heat tolerance, not a salinity tolerance, but heat tolerant. Then the other thing is that which I was talking about, the short duration window. If you have the window, very little window between two crops, you can grow the proso millet or finger millet. It's a 60 to 70 days crop only. So this is what I was thinking, that in case if we can introduce that one, and you can develop, because you can find in the supermarket, but it's not being grown in many of the countries. So those type of things that can be introduced and utilized as a in between two crops, in case if you want to exploit the more cropping intensity.
Very interesting. We have worked on the forage side, like some buffalo grass, panicum, some grasses. We have worked on really extreme conditions, but I don't know if we have worked on millet. And I think it's a really interesting to try.
Yeah, this is, I'm talking about the grain. Yes, I know that in the buffalo grass, etc. Actually, when you are having the salinity, you can grow for the buffalo grass, you can grow for the spirobolus, you can grow for the disticulus, you can grow for the another grasses. But those things can come only when you don't talk about the grain. It's a not food, but feed. I'm talking about here the food. So that is the one thing which I was thinking of.
Okay.
Okay.
Yes.
Okay, I think that we can ask some question to our programs. You know that INTA is organized in the research area as national program. Some of the national program are for value chain, and other national programs are for topics or thematic. So I don't know, if, Luis, if you have the answer, but if not, we can do some question to the coordinator of the program in order to know if we have something like is mentioned Rakesh.
Yes, we could ask of another coordinator, but I know that only in Amaranthus we work some we do some works with lines of Amaranthus, but we don't continue with this research.
Okay.
Okay, we took note of your proposal, so we will do the question in order to know if we have some capability or some activity in order to satisfy your question, Rakesh. Yes. Okay. Is it okay?
Yeah. Okay for me, because once we work together, we can go far.
Okay. Okay, so if you agree, we can close the meeting here. Yes. We can write a summary of the meeting. We will share the summary with our team, and after that with you, in order to agree with the final content of the summary. And if we can put one or two issues in order to work together in the future. And also we will do the question in order to know what another crops we can work. I don't know in the case of millet or other crops that we can include for the topic 2.
Sure.
Okay.
Okay.
I don't know if you or your colleagues, Rakesh,
Dr. Namo, Dr. Hebzur, if you have any question.
Another thing to say?
No, it's fine with me.
Okay. Luis, Diego, Luciana, Paola, ¿algo para agregar?
No,
I had a presentation. I'm sorry we didn't have time for this.
Oh,
But if you're interested in knowing our efforts in sorghum, I'm open to a new meeting if you want. We work on salinity and sorghum. I'm
okay.
sorry we didn't have time for my presentation.
Oh my God, you know, it is a missing. We miss your presentation.
No, if you have time, we can
Can
hear
you do it quickly, because I will be leaving 50 minutes is still there. So I
Okay,
can.
I can do it in 5 minutes if you would.
Yeah, please go ahead.
Okay.
Okay. Diego, ¿tenés para compartir ahí pantalla?
Yeah. Can you see it?
Yes, correct. I can see that.
Okay, I'll go fast. We work on grain, forage, and silage sorghum. We do some photosynthesis research. But I'm going to focus on salinity and alkalinity. This is the map of Argentina. These are the regions affected by these traits. So it's a really huge problem in our country. And we're working with a two-step strategy. We're working on mutagenesis. We worked on EMS-generated population for mutants, basically for selecting for germination. So we have selected in the field these mutants, and we have found some mutants that have up to 80% germination in really harsh conditions in salinity. You can see the conditions here on the left, 8.1 pH and 4.5 electrical conductivity for germination, which is a really susceptible stage in sorghum. And we are working now on hybrids derived from these mutants. The second part is working on some later stages salinity traits. We're working with some physiologists. So we have developed a hydroponic system. We have tested the system and it works for comparing genotypes. So now we are working on mapping, an association mapping study. So we want to go all the way to the genes or QTL affecting these traits in terms of growth and some photosynthetic traits. So that's basically what I mostly wanted to show. We also have collections in my research experimental station. I'm going to focus on sorghum collection. We have 2,866 entries, and we have characterized most of them. And we also have a core collection, a smaller sized part of the collection that is representing the diversity. And we have a lot of work on the collections as well. So just briefly what I wanted to show. Thanks.
Thank you. This is very interesting. So you have that the core collection, and you are using the core collection for association mapping panel, or
not that core collection. I am working on the association mapping collection developed by the USDA.
Okay.
Which is already genotyped and really useful for these kind of studies. I'm planning on adding our actual core collection, but it is not currently genotyped. So
okay.
we need funding for that. So that's why I'm using the other.
Okay. And then in Argentina, I can understand salinity, but do you have enough sodicity also? Alkalinity or sodicity you have?
Yeah, salinity and alkalinity, it's a huge problem in some big regions. Yeah, I think we're in the top 5 countries affected by salinity.
And then salinity I can understand, but sodicity or alkalinity, which you mentioned, this is pH of paste, or because 8 you put 8 point something, is it pH of paste, soil paste, or what?
Soil pH.
Soil paste, or pH 1:2. What is the like a unit you use for measuring the pH? Like, is it from the water, or it is the soil suspension, or it is the soil extract?
Soil. Yeah, it's soil extract. Yeah.
Soil extract. Yes.
Yeah.
Okay.
Yeah, we have a lot of regions that are affected by both conditions. So I expect the impact of these is going to be huge for recovering some lands affected by these. And yeah, the companies are not working on these for sorghum. And there's a huge interest in the farmers for this kind of.
And this sorghum is mostly for, because you
Forage.
said that for forage mostly, grains,
Yeah.
sorghum, forage and silage also, you put that, you know.
Yes,
So mostly
in
it is for
Argentina,
forage.
in Argentina, it's very similar to the US. It's mostly used for forage, not for human consumption.
Okay. And not even for sweet sorghum for the, like a biofuel, etc.
There was some interest some 10 years ago, 15 years ago, a huge interest in sweet sorghum for biofuel, but in the end, it didn't work very well. I don't know exactly what were the reasons, but I think it was economical, like it was not viable. Maybe Luis can expand because it was in Luis' region that the interest was.
Yes, I think that the reason is because sugarcane and maize or corn work better than sorghum for energy or bioenergy.
Okay. Okay, great.
Okay, it was fast. I'm sorry for that, but I wanted to show.
No, thank you very much. It's a very interesting, very interesting.
No, muchas gracias, Diego. La verdad que sí.
Muchas gracias.
Okay, thank you. Also, we will share with everyone the record of this meeting, so you will have there all the presentations.
Sure.
Okay. Thank you for your time, Rakesh and colleagues
Thank you very much, and have a
you.
good day.
Have a nice day. Bye-bye.
Thank you. Bye-bye.
Bye-bye.